everybody, and welcome to an introduction to mass spectrometry. My name's Antonia Wiesbicki, and I will be taking you through this today. So we'll cover the fundamentals of mass spectrometry, starting with single quad LC-MS, and then tandem quadrupole LC-MS, and then time-of-flight systems as well. And then we will talk a little bit about how to modify your LC method when adding MS detection. So let's begin then. When it comes to sample analysis, we're often tasked with answering one or more of these questions. What compounds are in the sample, so screening, how much is in the sample, quantification, what else is in the sample, and what is the difference between this sample and another one. And mass spectrometry is one of the techniques that we can use to do this. So what is mass spectrometry? Well, it's a technique for weighing molecules. We use an analytical technique that ionizes chemical species and measures their mass-to-charge ratio and displays this information as a mass spectrum, as can be seen below. What you can see is a mass spectrum with a mass-to-charge along the bottom, and along the side is the intensity of the ions to the detector. How are UV then and mass spec different? Some people in the audience may be used to UV detectors and trying to understand how mass spec is different. So with UV detectors, it contains a flow cell where light at a specific wavelength is directed through the sample, passing through the flow cell. And molecules that have a chromophore will absorb the UV light, and a detector will measure how much of that light is absorbed. A mass spectrometer, or sometimes referred to as mass detector, they ionize molecules, and these ions are separated by a mass analyzer and detected based on their mass-to-charge ratio. You can actually have a UV detector in line and then following that analysed by mass spec, so you can have the two detectors in line together. A mass spectrometer can be broken down into several constituent parts. So the ion source, where the components from the chromatograph are converted into gas phase ions. The mass analyzer, where ions are separated by their mass-to-charge ratio, and then the detector, where the different mass-to-charge ratio ions are recorded. And the data system follows, which acquires the data from the mass spectrometer, processes it, and generates the results. So the PC, basically. Most mass spec systems are defined by the mass analyzer that's involved, and the type of data you need may determine what type of mass spec system that you use. So first let's talk about the ion source. The process of converting our analytes and other components from the chromatograph into ions is critical to ensuring that they are detected. Several sample properties are very important when selecting an ionization technique. Analyte chemistry, the phase in which the sample enters the source, the polarity and molecular weight of the species of interest, as seen previously, can all be critical in choosing an appropriate technique. And for mixtures of analytes, the complexity of a sample may also play a role in the choice of what technique you would use. The chart in front of you shows the usable mass and polarity range for the different ionization techniques that are available that we will discuss during this session. So, for example, if your analyte is very small, very non-polar, there are a number of techniques that may work for you, and several factors can be considered. But if your analyte is quite large and quite polar, as you can see in this chart here, there may only be one technique available to you, which is ESI. This chart is by no means exhaustive, and a literature search may provide insights into the best technique for your analytes of interest. But for the purposes of today, we'll focus on electrospray ionization, or ESI, which covers a wide range of compounds, and APCI. For liquid chromatography, many modern mass spectrometers generate ions at atmospheric pressure. This requires that the source must not only create ions, but any solvent associated with them must be removed before they enter the mass spectrometer, and this process is known as desolvation. Gas and heat are applied in the ion source to ensure the desolvation process is very efficient, and proper desolvation is really critical to ensuring good sensitivity for the mass spectrometer. There are several atmospheric pressure ionization techniques used routinely in mass spectrometry, and we will look at three in particular today: ESI, APCI, and a little bit on Unispray. In APCI today, there's also Unispray as well. So electrospray ionization involves applying a voltage to a conductive capillary, typically made of stainless steel, and this applied voltage induces the liquid flowing through it to produce an electrically charged aerosol spray upon exiting. So the resulting spray then gives rise to ions contained in the aerosol droplets as they desolvate by interaction with heat and gas in the ion source. So the chemistry of your analytes is really important in ion formation in electrospray ionization. For basic compounds, like lidocaine, they protonate to form the positive N plus H ions. Acidic compounds, like ibuprofen, deprotonate to form negative M minus H or negative ions. So this is something to also consider when you're performing your analysis of your compounds with mass spectrometry. Electrospray is a solution-based process. The more ionizable a compound is in solution, the more likely they are to produce strong electrospray signals. The solvents that are used in liquid chromatography and the modifiers added to them will also impact the ionization, and we will examine this topic in more detail a bit later. So the soft ionization that occurs in electrospray can generate multiply charged species in both positive and negative modes. Since the species we observe are based on mass to charge ratio, the spacing of the 12C and 13C isotope peaks are separated by 1 mass unit for singly charged ions. For doubly charged ions, though, they are separated by 0.5 mass units. So essentially what you're seeing, as you can see in the slide here, is the N plus H is 1347, but the N plus 2H is half of that, so 674. Multiply charged positive ion generation also occurs in electrospray. As you can see from the horse heart myoglobin here, you can see multiple charges for this particular compounds where the molecular weight is 16,000, nearly 17,000 grams per mole. We can also see this when separating oligonucleotide species in negative ion modes. If we look at the mass spectrum for the 30 nucleotide species, we have observed from -8 to -14 charged states. So to summarise, electrospray is widely used because it can ionize a wide range of analytes over a wide mass range, from small molecules to large proteins. But as we saw earlier, it works best for compounds that are slightly polar to highly polar in nature. Just as an example of some typical electrospray positive ion samples versus electrospray negative ion samples can be seen in the slide below. If electrospray works better for polar molecules, then what can we use for the less polar analytes? And for those molecules, we can use atmospheric pressure chemical ionization, or APCI. So with this technique, ionization is achieved by passing the sample through a fused silica capillary. As it emerges from the capillary, it is nebulized by a flow of nitrogen gas into a heated tube where it is vaporized. Ionization is produced by applying a high voltage to a corona pin at the end of the heated tube. The two main mechanisms for APCI ionization have been outlined in detail in the paper referenced below by Horning et al. The first is a charge transfer mechanism, which is particularly useful for the analysis of non-polar compounds, and the second mechanism for ionization is by proton transfer, which is the main ionization pathway seen and associated with APCI in LC-MS. So here we can compare the two techniques in terms of ionization. the kind of analytes that work well, and the types of ions formed, and the LC flow rates which work best for each one. So many modern mass spectrometers can run both ESI and APCI with modifications with easily exchangeable source components, and some systems can run both techniques in tandem with the same chromatographic run. ESI and APCI, as we discussed, are used extensively, but there are other ionization techniques available. For example, the novel ionization source developed by Waters called Unispray. This is actually based on an ionization technique called the Coanda effect. More details of this can be found on our website. On this slide, you can see a difference in the response and signal to noise for Unispray, which is shown in red, and ESI, which is in blue, for 3 PFAS compounds. As you can see, there is a much higher response rate and signal to noise for the PFAS species in red compared to ESI, which shows one of the benefits of Unispray ionization. So now that we've created the ions in the source, we need to remove the solvent to ensure that we can detect them as we move from atmospheric pressure to the vacuum region of the mass spectrometer. The design of this desolvation region has an impact on the sampling of the ions created in the source. So while both the straight design and the off-axis design allow ions into the vacuum region, the off-axis design acts to reduce the amount of non-charged species and other background material that enters the mass spectrometer, which improves the sensitivity and the robustness of the instrument. So what happens at the sampling orifice of the mass spectrometer? So in this design here, there are 2 cones, the inner and the outer cone. The inner cone has a hole to allow ions to enter into the mass spectrometer, and the gap between the inner and outer cones in this design allows a countercurrent gas to pass into the source. And this helps with ion desolvation and to keep the non-ionized species away from the orifice to help keep them clean. And this allows more samples to be run on the mass spectrometer before cleaning the orifice is necessary. So once through the sampling orifice, some designs use a tube after the initial orifice to assist with further desolvation and to transfer the ions into the vacuum region of the mass spec system. So one downside of this design is that the tube could become blocked with non-ionic species from the sample and may require cleaning or replacement. Once the ions pass through the sampling orifice, there is typically another region with less than atmospheric pressure before ions pass into the vacuum region of the mass spectrometer. To ensure good transfer of ions through this region, a transfer optic is used. Depending on the system, this can be a multiple device like a hexapole or quadrupole, or a travelling wave device, and this transfer optic operates in what's known as RF only mode and does not do any mass selection. But this will come later. So to detect the ions, then that we've created in the ion source, we need a method to separate them. So this occurs in the mass analyzer. So once separated, we'll detect all our ions and determine their mass to charge ratio based on that measurement. Number of mass analyzers commercially available, but the most common types seen in laboratories are quadrupole-based systems, so either single or tandem quadrupole, which you may have heard of as triple quadrupole, and time-of-flight-based systems. There's also hybrid systems, which use some combination of these mass analyzers. There are other types of mass analyzers available, but we won't be discussing these today. Quadrupole-based systems. And let's discuss how quadrupoles work. So a quadrupole is an assembly of 4 parallel rods arranged equidistantly from a central axis. Through the application of RF and DC voltages, ions can be filtered along the central axis and their mass measured to yield a spectrum. So depending on the exact potential applied to the quadrupole, ions with masses too large or too small will not pass through the quadrupole, and these ions will strike the rods and they'll be lost. So ions that are generated in the source pass through the transfer ion optics, which focuses the beam to enter the quadrupole. The ion beam is likely to be a mixture of chemical species with different mass-to-charge ratios. So for example, here we have ions with mass-to-charge 220, 310 and 400. The combination of the RF and DC voltages are applied to the quadrupole to create a fluctuating field, which will only allow one specific mass-to-charge ratio to pass through. All other ions with different mass-to-charge ratios will strike the quads, become discharged, and the vacuum system will pump them away to waste. So this is how the mass spectrometer is filtering the ions to through the quadrupole. So in this example, as we've shown in the previous slide, what you find is that the mass charge of 310 is transmitted, and the purple and the red, so 220 and 400 ions, are removed and pumped away. So the mass spectrometrist can actually tell the quadrupole to pass the mass of interest or scan a mass range via the software control. And this software adjusts the RF and DC voltages applied to the quadrupole during the experiment. So while it's very easy to use a mass spectrometer, what's going on within the software is much more complicated. So let's talk about the types of quadrupole-based systems now we understand how a quadrupole works. So we have single quadrupole and tandem quadrupole. And with the two techniques, you can do full scan acquisitions and single ion recording. With a tandem quadrupole, you can then do a few more different experiments as well. So here is an example of an MS scan. An MS scan is ideal for screening methods where the user is looking at everything in the sample. In an MS scan, the transfer optics which move the ions from the ion source to the quadrupole run in RF only mode, meaning they transmit all the ions from the source to the quadrupole. The voltages on the quadrupole, so the second mass analyzer, are scanning, transmitting ions into the sequence to the detector. Once the instrument has passed through the mass range of interest, it repeats. And this can be done really, really quickly, allowing the user to acquire a scan in milliseconds. And this produces what's known as a total ion chromatogram, or TIC. So for example, we may be scanning the mass to charge range of 200 to 1500, just as an example. Here is an example of the data collected from a single quadrupole in full scan mode from isoflavones in a dietary supplement. The data was collected in Electrospray positive mode. If we look at the stacked plot of UV spectrum at 260 nanometers compared to a total ion chromatogram TIC collected in Electrospray ionization positive mode ESI, one thing that we observe is the difference in intensities of the various peaks. In the TIC, there appear to be many more peaks, and this is due to the higher sensitivity of the mass spec detector. In addition, some of the larger peaks are in different ratios, and that is because chemical species can have very different ionization efficiencies compared to each other. So relative response of analytes varies in each technique, and the y-axis equals the counts in MS versus the AU absorbance units in UV. You can also use a single quad mass spec to do single ion recording or single ion mode, and this fixes the quadrupole voltages so that only a single mass to charge ratio is transmitted to the detector and all other ions are barred from passing through the quadrupole. It's a really way to optimize the sensitivity of your specific analyte of interest, and it provides a high sensitivity because the dwell time is spent in a single mass to charge and is most commonly used for quantitative analysis. So you can set the SIR mode to, say, for master charge 459.0 and 453.1. You can do both in one run as well. And here's just a quick example of SIR data from a single quad mass spec. Each SIR channel represents a single master charge value, and it can be collected over the entire sample run, or it can be collected for a portion of the runtime in Windows, as we can see here. And this provides maximum sensitivity of each of the compounds of interest. So they're the two single quad modes that you can use, which were scanning and single ion recording. So let's look at tandem quadrupole mass spectrometry now. So tandem quads have two mass analyzers. So the ions generated in the source are filtered by the first mass analyzer. And then in the middle there is a collision cell, and this is used to fragment the molecules exiting the first mass analyzer. So that's the first quadrupole. These fragments are then filtered by a second quadrupole or second mass analyzer. And the selectivity of the tandem quadrupole systems has made them the industry standard for mass-based quantitation. So just to summarise, and some people will call tandem quadrupole systems triple quad systems. I tend to do that as well. It doesn't mean that the mass spec actually has 3 quadrupoles. It actually only has 2 quadrupoles, and in the middle it has a collision cell. And that's why tandem mass spec is also used to describe this technique, and it's probably more accurate. So again, with a tandem quadrupole, as with a single quadrupole, you can do full scan acquisitions and single line recording, but you can also do MS experiments too. And we'll talk about a few of those now. We've got product ion scanning, precursor ion scanning, constant neutral loss scanning, and multiple reaction monitoring. Product ion analysis involves fragmenting a known precursor ion and scanning a range of product ion masses to observe the effect of fragmentation, and this is the traditional MS experiment. So the voltages on the first quadrupole are static, passing a selected ion of interest into the collision cell, and a voltage is applied in the collision cell to accelerate the ions into the collision gas. It's typically usually argon that's the gas of use in collision gas cells, and it results in fragmentation of that particular ion. The ions from the fragmentation will then pass into the second quadrupole, which is scanned to produce the mass spectrum. A product ion analysis is selective because the product ions in the spectrum can only originate from fragmentation of a specified precursor ion, and this mode of scanning is used to gain structural information or to identify product ions for use in creating MRM transitions. This slide shows a product ion or MS spectra of chlorpheniramine. It has a precursor ion at master charge 275, and its product ions are lower in mass than the precursor. If we adjust the collision energy applied in a collision cell, we can affect the distribution of the product ions, and as we increase the collision energy, we create lower mass product ions, and this could mean that we are losing product ions which are indicative of a molecule structure like the ion at master charge 230 that you can see in all of the chromatograms there. The product ion spectrum can become complicated when dealing with multiply charged precursor ions. In the case of the antibiotic peptide vancomycin, as can be seen here, the precursor ion is doubly charged ion at master charge 725.6. When it's fragmented in the collision cell, though, the products are singly charged, and so they can occur at a master charge value that's higher and also lower than their precursor ion, so it can be a little bit more complex. Let's think about precursor ion scanning then, sometimes known as parent scan. So precursor ion analysis is used to determine which precursor masses have similar fragment ion. So the first quad is scanned to pass a range of ions to the collision cell for fragmentation, and then the second quad isolates a specific fragment ion from the fragmentation in the collision cell. Precursor ions pass through the first quadrupole that produces a product ion at the specified mass of fragmentation in the collision cell, registers a response at the detector, and the mass of the precursor ion is then shown in the spectrum. So precursor ion analysis is selective because only precursor ions that produce the specified product ion are observed. And this type of scanning is used to confirm structural information produced by product ion analysis, as well as for screening for groups of compounds with common structural features. Here is an example of what I mean with 3 steroids, so hydroxyprogesterone, progesterone and testosterone, whose precursor ions all fragment to create a product ion at master charge 97. If we examine at a product ion scan of hydroxyprogesterone, we can see the master charge 97 fragments. If you look at the precursor scan of master charge 97 for a mixture of these three steroids, we can see the precursors that generate the product ion. In this case, we see testosterone at master charge 289.4 and progesterone at 315.4 and hydroxyprogesterone at 331.5. So you can really see a difference between these three compounds with this type of analysis. Neutral loss analysis is another option as well, and this is useful in determining which compounds have a similar fragmentation pattern. Mass spectrometers require a species to be charged in order to be detected, so if a neutral fragment of a molecule hits the detector, you won't see it. However, we can look for the result of that neutral loss by operating both quadrupoles in scanning mode with an offset between the two, and that offset will be the neutral loss that we're actually looking for. So here, the two tricyclic antidepressants, trimipramine and desipramine, produce a common fragment at a molecular weight 195, but it is not charged, so it will not be observed by the detector in the mass spectrometer. However, the charge fragments for each molecule will be observed. In the constant neutral loss spectrum, we can see the precursor masses that undergo a loss of 195. Multiple reaction monitoring is a non-scanning mode used for high-sensitivity analysis of target analytes. So it is a primary method of data recording used by tandem quadrupole mass spectrometers for quantitative analysis. In this experiment, both quadrupoles allow only transmission of the specific precursor ion that provides a specific product ion for monitoring, so it's very, very targeted, it's very selective and very sensitive, because only a specific ion, which fragments to produce the specific fragmentation ion, is monitored for the entire data acquisition time cycle, rather than just a portion of the cycle in the case of a full scan. So an individual MRM does not provide information about other ions in the sample, as you would see when you're scanning over a mass range, but it's very similar to a single ion recording experiment or SIR experiment with a single quad, but this is where you're looking at the parent to daughter or precursor to product ion scanning. And it can be multiple MRMs can be run either consecutively or concurrently with others to look for multiple species in a sample if that's required. This is quite a nice example of two NSAID or non-steroidal anti-inflammatory drugs, ketoprofen and fenbrufen. On the left-hand side, we have a sample that contains 60 nanograms per mil of ketoprofen and 60 nanograms per mil of fenbrufen. And while they're not completely separated, we can see two peaks in the single ion recording chromatogram at 255 master charge. On the right-hand side, the sample has 60 nanograms per mil ketoprofen and 6 nanograms per mil fenbrufen. And it appears that we can still see both compounds, but can we be sure that that shoulder is actually fenbrufen and that the shoulder isn't hiding in the left-hand chromatogram? The SIR chromatogram has a shoulder, but if we run an MRM experiment on a TQ or tandem quadrupole mass spectrometer, then we can use the additional specificity of the MRM to look at different MRM channels for the two compounds. So both compounds do have a precursor mass of 255, but ketoprofen has a product at 209 and fenbrufen at 237, so they can be differentiated between the two when using MRM mode compared to SIR. And this is how the specificity of MRM mode can really help when looking at isobaric compounds. Here's a diagram of how time-of-flight mass spectrometers work. So this is a very different way of analysing the compounds compared to the quad systems. So think of it like a 100 metres track race at the Olympics. When the gun goes off, the clock is started and the three molecules, lightest in red to heaviest in blue, start from the same place and are given the same amount of energy by a voltage pulse applied to the pusher. They then separate as a function of their mass to charge, meaning that the lightest, so the red species, arrives at the detector first, followed by the green and then by the blue. As they hit the detector, their time of flight is recorded, and that time of flight is proportional to the ion's mass to charge ratio. The mass to charge ratio is calculated by the data system and is used to generate the observed mass spectrum. In this ideal example, all of the ions of the same mass to charge would arrive at the same time, and this would mean all our spectra would have very narrow mass to charge peaks. In MS terminology, this would give our mass spectrometer incredibly high resolution, something that is always of interest to mass spectrometrists. But what do we even mean by resolution in mass spectrometry? Mass resolution typically refers to the ability to separate two mass spectral peaks and is calculated by dividing the mass by the peak width. Sounds complicated, so let's go through it. So, time of flight mass spectrometry is one of the MS techniques considered to have high resolution. The black peak here shows melittin at a resolution of 1000, which is a level of mass resolution routinely observed on a quadrupole-based mass spec instrument. Now, the red spectrum shows the same melittin with a mass resolution of 10,000, a resolution that is easily achieved by modern time-of-flight mass spectra systems. With a high resolution, analysts are able to observe all of the isotope peaks of melittin, which cannot be observed on a quadrupole-based system. But what experiments then can be run on a time-of-flight mass spec system? The most common MS data acquisition mode for TOF is full-scan MS, where the entire mass range of interest is acquired through the LC experiments. From that full-scan data, masses of interest can be extracted during the data analysis to form extracted ion chromatograms, or XICs. Hybrid mass spec systems using time of flight, such as QTOFs, are also capable of MS over MS analysis. This type of experiment isolates a species of interest or precursor within the mass spectrometer, and then it is fragmented. The product ions from this fragmentation process are then recorded in the mass spectra on the data system. Some modern TOF systems are capable of running an MRM-like experiment, allowing for both high resolution, high mass accuracy data and quantitation to be run on the same system if necessary. Many TOF MS systems also have other modes of data acquisition that are used for specific applications, where both MS and MS-MS data can be acquired in the same LC run. These methods can be separated into two groups, DDA and DIA. Let's talk about these two. So DDA is data-dependent analysis, which begins in mass spec mode and where certain criteria are met, such as specific masses, charge states, or intensities. The MS switches to the MS-MS mode and the product ions are recorded until another set of criteria are met. At that point, the system switches back to MS mode and looks for other ions to meet the criteria. Data-independent analysis, on the other hand, collects data on precursors, as we've talked about before in the quadrupole section, and products, so precursors and products throughout the LC-MS run, typically by alternating between MS and MS-MS-like modes. And the data system then uses algorithms to assign product ions to the correct precursor ions. In time of flight, the TOF system cannot physically filter the individual ions, so the TOF system records all the ions at the detector, so some of the experiments run on quad-based MS systems are not available on a TOF system, for example, neutral loss or MRM mode. But some modern TOF systems are capable of running an MRM-like experiment by only recording masses of interest and discarding the other masses, allowing both for high resolution and high mass accuracy data, and also quantitation to be run on the same system if necessary. Just wanted to show as well that if we make the time of flight longer then we can get more mass resolution, with many TOF systems using orthogonal extraction to push the ions as they enter the TOF region of the instrument. This allows us to couple a continuous ion beam with the inherently pushed technique of time-of-flight mass spec. We can increase the flight path length to increase the mass resolution by using a reflectron, which sends the ions on a V-shaped path to the detector. You can even have a dual reflecton, which is a W-shaped flight path, and you can even have multi-reflecting systems, which have multiple reflectrons, making the path length much, much longer. This is similar to our Zevo MRT and our MRT systems. So once we have separated or isolated our ions of interest using a mass analyzer, we need a method that can detect them. So let's talk about the detectors. There are two common detector types used in mass spectrometry, so electron multipliers and photomultipliers. There's also microchannel plates as well. It's good to be aware of. So in a electron multiplier detector, the ions formed on the mass analyzer and strike the anode surface and electrons are ejected. And electron multipliers are the most common detector in mass spec systems. They're very sensitive and have a fast response, but in many systems they need to be replaced every few years. In photomultiplier detectors, which is an alternative option, ions strike a conversion diode and generate electrons, and these strike a phosphor screen, and the phosphor emits photons. Keeping the photomultiplier permanently under vacuum prevents contamination and allows the detector to maintain its performance for considerably long periods, longer than the conventional electron multiplier before. This detector is used in nearly all of water's quadrupole-based mass spec systems. A third detector that you can have is a microchannel plate, which is basically an assembly of single point detectors that are all connected together and act as a single detector, kind of like buckets on the floor when you're trying to collect rainwater, say. As ions hit the detector, they produce electrons, and these cascade through the individual electron multipliers, creating secondary electrons within the wall collision, and this creation of more electrons amplifies the signal. The data system that receives this signal and converts it then to a mass spectrum. If this seems quite complicated to you with the different detectors, what I would say is don't worry about it too much. The computer that controls the mass spec will deliver the information you need, and you don't need to worry too much about those things. They're just small differences. So we've covered everything now, from sample introduction to ion source to the mass analyzer and the detector and the data system, as you know, you interact with every day. So the ion source, where the analytes are converted into the gas phase ion, the mass analyzer, where the ions are separated by their mass to charge ratio, and the detector, where the different mass to charge ratio ions are detected. So let's just quickly compare the mass analyzers together. So we're thinking about mass accuracy quickly. So time of flight systems are typically accurate to the low ppm levels, even with fast chromatography. Quad systems are considered nominal mass instruments with a mass accuracy of plus or minus 1 Dal. With a quad instrument, as we've seen before, you can get 37 possible results from one particular nominal mass. So how does this compare to time of flight? So with a time of flight, systems are capable of exact mass measurements to a few ppm, and this means that the accuracy of the mass measurement allows us to tighten the mass tolerance much higher. So we only have 3 possibilities, as we've gone from 50 ppm to 3 ppm. And so using the C, H, N, O and S, or carbon, nitrogen, hydrogen, oxygen and sulfur, we can only have 3 possible results that it could be. So if we're working with unknown compounds, we should feel very comfortable in saying that the peak is consistent with the composition that we've stated here. But to learn more about the structure, we would probably need to perform other experiments such as MS acquisition to obtain more information about that structure and the fragmentation pattern of that structure too. The ability to quickly acquire good quality mass spectral data is an important factor and will be related to the type and speed of chromatography being coupled to the mass spectrometry system. Time-of-flight systems record all of the mass range being acquired all of the time, which can give these systems an advantage over systems which scan the mass range. Quad-based systems have to change the voltages as they scan the mass range, which can slow acquisition rates for full-scan MS. Acquiring faster or wider mass ranges can lower the time spent on each individual mass, meaning lower quality data could be acquired. Total cycle times for ion trap systems are comprised of multifunctional stages and can vary significantly based on the desired mass analysis times and selected mass ranges. So as we can see, adding a mass spectrometer to your system can add a lot more information to your assays, but there are some things to be considered when you add an MS to your detection options with an LC. Many ways to connect an MS to an LC system. Each has its own benefits and disadvantages, so to decide on what type of system configuration we need to achieve our experimental goals, we need to think about the type of analysis that we need to run. We will also need to think about which MS system meets our needs and the performance capabilities of the MS systems available to us. So all of these factors will impact how we configure our system. When we add MS to an LC-UV system, we must consider the principles of ionization that we discussed earlier, and as we talked about as well earlier, that we need the analytes to be ionized to be observed in the mass spectrometer, and this means that the mobile phases must be volatile as well to allow for the dissolvation of the analytes. So this is what happens if you add non-volatile additives to your mobile phase and run it through a mass spectrometer. It actually forms these solids within the source, which is definitely not a good idea because you will lose sensitivity and won't get the results that you need. So this is why we tend to advise that there are some unsuitable additives for all MS systems. Non-volatile salts like phosphate, borate and similar can deposit in source and plug capillaries, meaning more cleaning or maintenance may be required. Orthogonal spray surfaces, such as Z-spray sources, can handle non-volatile additives without plugging, but frequent maintenance may still be required. Surface active agents, surfactants and detergents, can suppress electrospray ionization. So examples of that include STS or non-aryl sulfate, and inorganic acids like HNO3, HCl, hydrosulfuric acid are corrosive and can corrode the metal of the source and can potentially damage it and its associated electrical components. The most efficient conversion of electrospray droplet charge to gas phase ions, that is, the best electrospray sensitivity, comes from small, highly charged droplets. Increasing the organic content will decrease the surface tension and increase the volatility of the mobile phase, both of which lead to the formation of smaller droplets. In this regard, organic solvents are superior to water for ESI. When using higher percentages of water, it is usually necessary to increase the temperature in the source to aid dissolvation for both ESI and APCI. In negative ion electrospray, lower electrospray capillary voltages must be used to prevent corona discharge at the tip of the capillary. Substituting electron scavenging solvents such as propranolol, isopropranolol and butanol for methanol and acetonitrile may permit operation at higher negative voltages without creating a corona discharge. An additive can be added to assist with ionization in both positive and negative ionization modes, but many should only be used in low concentrations and with caution, as we'll see on the next slide. Although many of these additives can be used with MS and are compatible, some should be used with a bit of discretion, and that includes TFA, which can suppress negative ionization and to a lower extent suppresses positive ionization as well. Triethylamine also can suppress ionization, and this is more likely to occur in positive ESI. Lastly, some mobile phases like THF and normal phase solvents may not be compatible with all of the components in the mass spec. This example shows how additives in a mobile phase can impact ionization. So in this example, some ionization occurs with no additive in the presence of acetonitrile and water. But if we add formic acid, just a really small amount, then we can see a significant improvement in ionization. But conversely, if we were to add 0.1% TFA, we get suppression of the analytes is much smaller. So we do have to be careful when we select our additive in the mobile phase, that you will see. pH response is also a consideration, so high or low pH can improve sensitivity. So you might want to consider the pKa of your analyte. This is obviously something that you consider anyway when developing an LC method, so not so different from mass spec. And as you can see, the impact of pH on MS sensitivity can be quite important. So here you've got the peak of signal to noise is 5,000 roughly, and if you add a high pH, you can double almost the signal to noise of that particular peak, which is really, really good. Another aspect to consider is just the flow as well. So if you've got a HPLC system, the flow rate may be quite high, maybe 1 ml per minute. So you just want to make sure that you don't flood the mass spec with too high flow rates. This can be a two-pronged issue. So you might end up with too much liquid in the mass spec, causing flooding. But you could also maybe overload the detector with the amount of sample you're adding, because mass spec's much more sensitive than HPLC UV. You also might want to consider, if you're adding a mass spec, the optical detector flow cell back pressure. There will be a slightly higher pressure if that flow from the UV detector is going to a mass spec compared to if it was just going to waste. So another thing to actually consider as well. But you can speak to your manufacturer or your Waters representative to understand how that might affect your data. This is an example of what it looks like when there is oversaturation in the mass spec, or when you put too much into the mass spec. So in this example, the UV detector does not appear to be oversaturated, but when the sample is observed under the MS, the peaks are all flat-topped, which indicates oversaturation, or hitting the roof, as we used to call it in my old lab. This is not unexpected because the MS is more sensitive than the UV. However, we need a way to reduce the oversaturation in MS and align the responses in both UV and MS. So if the full flow poses issues for your system or assay, the next option is to split the flow between the two detectors. This configuration uses a splitter so that the UV and MS detectors can be run in parallel. So this configuration requires additional tubing and a T, but it reduces the chances of oversaturation of the mass spec detector. And in addition, it can allow for better control of the material actually going into the mass spec. However, it could be difficult to optimise the split flow ratio, and the response may be lower in the UV since all of the sample is not actually going to the UV detector. This slide shows the impact that a splitter can have on the calibration curve of an analyte. And in this example, terphenadine is shown from 0.1 to 50 ppm on two system configurations, one without a splitter and one with, and you can see the more linear response of the MS signal when the splitter is used. So based on the tubing internal diameters, we can determine the approximate split flow ratio for the splitter. And we can also adjust the tubing length and or the ID to change the ratio. There are online calculators to assist with this calculation type. And if you don't want everything that elutes from your LC to go into the mass spec, you can use what's called a divert valve. And this can be helpful to divert anything that might contaminate your mass spec, like salts or column wash, and ensure the best possible MS sensitivity all the time. So the configuration has a divert valve that is plumbed between the UV and the mass spec detectors. It requires the control of the divert valve in the instrument software, and for the timing of the valve to be switching to be programmed in the method. It's also worth remembering that divert valve does not mean that the system is compatible with the non-volatile buffers. You'd still have precipitation in the MS source during that portion of the run if it's not diverted away from the mass spec. You will also need to use low volume tubing that will reduce the extra column dispersion into the mass spec to ensure good peak shape and analyte sensitivity. And this is just the last example in which the divert valve has been used to keep the source clean and reduce carryover. It's an example with an API and its related impurities. It is the impurities that we're interested in, and the API is at a much higher concentration and has poor peak shape. So having all that API material going to the mass spec can cause contamination of the source and overall affect sensitivity. So using a divert valve to divert the API away from the mass spec means the impurities elute as expected and there's no impact on their peak shape. This allows for us to keep the source clean and continue to obtain the best MS sensitivity possible for compounds of interest. So in conclusion, mass spectrometry is a powerful and sensitive detection technique that can bring significant benefits to your assays. There's multiple different types of MS systems, and your choice of MS technique is based on the goals of your analysis. When you add MS detection to an existing assay, or when you're building a new assay, the LC method conditions would need to factor in the ionization methods of ionization needs of your MS systems. You may need to adjust existing methods to ensure best sensitivity for all your compounds. And when you add MS detection, you need to consider how you might set up the MS in combination with existing detectors to ensure best sensitivity and also maximum uptime for your system.